Happy New Year and welcome back. As you can see, we're going to begin our semester with genetic engineering. And here we have these pictures of these awesome glowing cats. To find out how they're glowing, go ahead and read this portion down here about the proteins and such involved. Now, it is important as we start this to know that scientists can't just come up with this wild and crazy idea such as, ooh, can I make a cat glow, and do it. They've got to have a reason behind it. What's the purpose? And how is this going to benefit society later on? And so in starting this unit, you're able to get some bonus points. Google, or otherwise look up, on Bing or whatever else, and you can get some bonus points on telling me why they made these cats glow. What was the actual purpose? So let's begin. And we're going to start with selective breeding. Now, selective breeding, this is a type of genetic engineering. However, there's not a whole lot of manipulation of genes involved. It is simply breeding. It is taking organisms with desired characteristics, what do we want, and producing the next generation. And I'll give you a couple examples of where that has happened. The first one being these bell peppers. Usually people like the sweetness of these red bell peppers. However, they're a little small. So you can see here, this yellow one is a bit larger. So they've taken this yellow one, bred it with the red one in order to get this desired hybrid of a larger red bell pepper. And here we get different colors of cauliflower. I personally haven't seen any of these, but I know that they do exist, and scientists have done this for the levels of vitamins. You get more vitamins out of these. The wolves and the dogs. It is said that all breeds of dogs, including that little Chihuahua, has descended from wolves. And man, over many, many centuries, has selectively bred dogs for different traits that are desirable. For example, we get the Labradoodle. This was produced or selectively bred by crossing a lab and a poodle. Why? Well, labs are great dogs. They're super friendly, nice. They can be protective. I love my labs, but they do shed a lot, and they are kind of smelly. So not even scientists, but people said, okay, we would still like the lab and the personality, but we don't want the shedding issue. So poodles don't shed much. They cross them, and we get this dog here, which is a cross between a lab with the nice personality and the less shedding. Hybridization. This is a type of selective breeding. We're going to take two dissimilar individuals and bring the best of both worlds together. For example, here we have the Brahma cow. You find these in Texas a lot. You can tell the Brahmas by the big hump on their back. And they are very, very good in the heat, but the meat on them is just really not high quality. And so we look at the Angus cow, which has a high quality meat. The farmers took these two cows together and they get this nice Brangus cow who has good meat and can withstand the heat of Texas and Arizona. Luther Burbank, he was very, very important in hybridization, especially with plants and such. And you'll see here that he selectively bred plants and developed the disease-resistant potato, which was really good because about his time, if you remember in history, that Irish potato famine, which really wiped out a lot of potato crops around the world. He also developed this Shasta daisy. It is fuller and more beautiful than a regular daisy. Now put this in your notes somewhere, that Luther Burbank actually developed over 800 hybrids. He worked a lot with plants and developed over 800 of them. Here we have the broccoli flower. This is a cross between cauliflower and broccoli. Milder, slightly sweeter flavor than both the broccoli and the cauliflower. And here we get a few of them. Totally adorable, right? Here we have the lion and the tiger, and we end up getting from those, if the father is a lion, we're going to go ahead and from that lion father and the mother tiger, we're going to get a liger. However, if the father is a tiger and the mother is a lion, we're going to get a tigon. And then this adorable zonkey, a zebra and a donkey. Now with these types of hybrids and such, This is not something that would happen in nature, and this is more of a forced issue, and generally they are sterile. So these Tigons could not go on and breed and make more Tigons because of the sterility. And the sterility happens because lions and tigers actually have a different number of chromosomes naturally, and so it just kind of messes things up, and they would not breed. Like, we're not going to have zonkies breeding with zonkies to produce more. Inbreeding, okay, go ahead and get the giggles out, but inbreeding is an issue, and it's basically the continued breeding of individuals with similar characteristics. This will ensure that the characteristics that make each breed unique are preserved. Okay, we like having some of these characteristics. You look at this cute little puppy here. Purebred dogs, lots of inbreeding can go on in order to keep those traits of those dogs there. Serious problems, two recessive alleles can come together, and that's where we oftentimes get the albinos. Now in the wild, albinos are very, very much not so common because they stand out and would be kind of available to predators more readily. Okay, so increased variation in a population, mutations. We're going to get more of a variety of things in a population if we induce mutations. Now, mutations we often think of as being very, very bad. That's our sci-fi movies coming in telling us, oh no, we've got mutations and you have these weird things. But mutations can actually be very, very good. Polyploidy, there's that polyploidy. Remember, poly means many. Ploidy, remember in genetics when we had our diploids and our haploids. Diploids was where we had 2 chromosomes, and haploids half the number where we had 1. Poly means many. Now, in humans, and then primarily in animals, if you get more than 2 chromosomes, if you get a polyploidy, usually that's very dangerous. For example, trisomy 21. You get 3 chromosomes of number 21. That's going to be Down syndrome. It can be detrimental. But in plants, polyploidy is pretty awesome because you get more robust plants and such. Bigger, fuller, and that helps increase our food supply. Okay. To introduce the mutations, a lot of times they use radiation or chemicals. Oil-eating bacteria. This has been a wonderful mutation come about. Bacteria that eats oil. Think of the oil spill that happened a couple years ago in the Gulf of Mexico. It was detrimental, of course, especially at first. However, they introduced into that area this oil-eating bacteria, and that really helped decrease the oil in the area very, very fast. And we don't really hear about the detriments of it anymore. And that's because of this bacteria that ate up all that oil and made it very good for us. So here's a bit of review. On your papers, go ahead and put A, B, C, or D. This is question 1. If this goes too fast for you, you always have the pause button. Question 2. Question 3. Question 4. And question 5, you're going to go ahead and put the 3 words that would go in those 3 blanks. And here we begin the next part, 13.2, manipulating DNA. So here's the part in genetic engineering where things are actually changed by a scientist or used by a scientist. Okay, it's not just the breeding anymore. So here's the primary definition of what's going to be happening here. Scientists use their knowledge of the structure of DNA and its chemical properties to study and change DNA molecules. Both words are important there. Sometimes it's a studying, sometimes it's an actual change. Okay, different techniques. We've got different things that they can use to extract DNA from cells. We did that in class. Look at this picture here, and remember when we did that with I think we used bananas, and we were actually able to see that DNA. They can cut DNA into smaller pieces. That is very, very useful, like in DNA fingerprinting, which tomorrow we'll do an activity in class to show how that is done. This is what they do in forensics a lot to catch criminals or figure out who the actual father is of the baby, paternity tests. They use different techniques to identify the sequence of bases, what order do they go in, and separate DNA fragments. Cutting the DNA. Yes, you can actually cut it, even though it's very difficult to see the actual things going on in the DNA. But we've got these things called restriction enzymes. And they can cut DNA in precise nucleotide sequences. Remember the nucleotides being that A, T, C, G. So if we get a sequence that might be A, T, C, G, and whenever that came up in a whole line of DNA, the restriction enzyme would cut it at that point. So it's kind of a trigger of where can these be cut. The RE stands for restriction enzyme, cuts the DNA sequence only if it matches the sequence precisely. So identifying the base sequences. Here, in this picture, what you see is like a DNA fingerprint type thing. These are, it's called gel electrophoresis. Whoops. And it's going to use electricity to separate pieces of DNA based on their size. So the restriction enzymes are used to cut the DNA, and then this gel electrophoresis zaps in electricity, and it will actually separate the pieces so that scientists or forensic persons and such can compare them to others. So a mixture of DNA fragments is placed at one end of the porous gel, and electric voltage is applied to the gel. Negatively charged particles move towards the positive end, and the smaller parts move furthest. Smaller probably because they're less dense. And here is a picture just kind of showing what happens. So the restriction enzymes have taken this DNA here and cut it into these pieces. The DNA is poured into these little tiny wells in a gel, and they're going to be separated. The negative, when we've put it up here, everything's going to move, and you can read each thing that goes there to see how it is done. Okay, so go ahead and take a minute to read the cartoon. I think it's adorable. But genetic engineering, the actual definition, and there's a part on your page where I do want you to put the definition, making changes in DNA code of a living organism. So we're actually inducing that change in the DNA, and that's what genetic engineering here. So using DNA sequences, study specific genes, compare, so scientists are going to study them, compare the genes with other organisms, and discover the functions of different genes and gene combinations. So we can actually see what this combination of genes would do and such like that. Because remember, genes are made up of those proteins, and those proteins each have different functions. And so scientists have been looking at these sequences in order to figure out what each does, and they can use what they do in order to change things the way they changed. So the techniques to do that, they're first going to put in the unknown DNA in a test tube. DNA polymerase, remember that from before break, that is added and it makes copies of the strand. So remember, the DNA polymerase is what actually makes the DNA duplicate, and that's what's happening here, it's duplicating. Then add bases with chemical dye attached. So if we have base A, it's going to be maybe red, base T, maybe blue. And so that dye is actually going to help read things. Each time dye-labeled base is added to new DNA strand, the synthesis, remember, synthesis is putting things together, is terminated. So the new strands are actually going to be different lengths. The dye-tagged DNA fragments are separated by gel electrophoresis. Okay, and here you can see the blue they used for the G, the red they used for C, green A, pink is T, and they have, and this is to help them determine the sequence of DNA bases and such. So then recombinant DNA, recombinant, recombine. DNA produced by combining DNA from different sources. So they're recombining it from different sources. So you'll see here the restriction enzyme cuts both DNA strands at the same site. So you've got here a piece of DNA and then a piece of foreign DNA here, and the restriction enzyme has cut them both. And in here, the restriction enzyme used, we can see the TTAA, and you've got the same thing here, just going backwards, it's just flipped, okay? And then they're going to be put together. So then we get the PCR, polymerase chain reaction. DNA is heated to separate the strands. So we have here, you can follow the flow chart, 95 degrees. So you've got the two things of DNA here. It's heated, and we get one strand here, one strand here. Cooled, and a lot of these primers, the primers being this green and this pink, kind of adds on to them. And then DNA synthesis is being put together in order to start making copies. So it's a chain reaction, and you can see once we've got that, we can do it again and again. So here's your review. And this should be A, B, C, D. Oh, here's the letters over here on this one. Oh, I guess the letters are off to the side. When I download these, sometimes the formatting gets a little bit messed up. So this is question 3. And that is the end there. You'll see you do have another, you're doing a thinking map to go with this to show the flow of how this DNA reading and such happens. And in class tomorrow, what we're going to do is go ahead and work with, it's going to be paper, but it'll show you more of that DNA fingerprinting and how maybe a criminologist or a forensic scientist or somebody determining the paternity of a baby would happen.